Greetings. My name is Dr. Liz Pierce, and it is my pleasure to welcome all our guests to this week's edition of our Integrating Bioinformatics Education Series. Today, I'm pleased to welcome Dr. David Donley. Dr. Donley is an assistant professor in the Harding University's Department of Biology. By training, Dr. Donley is a neuroscientist and molecular biologist with research interests in neurological diseases and neuroinflammation. In today's session, Dr. Donley will discuss how his lab is using proteomic data to involve undergraduate students in research. He will describe multiple approaches for using large data sets to promote student engagement in research experiences. His talk will also discuss how to reduce barriers to entry for undergraduates and how to help students gain confidence in computational strategies. Dr. Donley, thank you very much for talking with us today. We look forward to your presentation.
Thanks, Liz. I'm looking forward to sharing my approach to this, and I will say, by training, I'm not a bioinformatician, and so a lot of this has been experiences that I've had that hopefully stimulate some ideas, some discussion as we go along. So before I get started, let me first say, if my screen will switch, I have had a lot of help along the way. I've had some fantastic students. Today, I'm going to talk about some work primarily from Landon and Allison, two of my students. Landon right now is a PhD student in Rhode Island, and Allison is one of my current students. And I've also had a lot of help from the UAMS Bioinformatics Core and learning all of this with some of my data. And then I'd also like to thank my funding sources, and especially the Arkansas Embry, who's been very supportive of my ideas and my work so far. So what I want to talk to you today about is how do we begin at the beginning and then keep going until we get to the end. And so this is a quote from Lewis Carroll in the movie The Mad Hatter says it. In the book, I think The King says it. And the question that I want to think about is, what does that mean for bioinformatics education at the undergraduate level? And this can be a tough question because undergraduates need both the knowledge of computer science and the tools to do this, as well as the biology knowledge. And so what I want to think about is, how do we actually start to incorporate some of these ideas and get them comfortable in a way that reduces some of the mystery and the daunting task of beginning. And I think a lot of times undergraduates view bioinformatics and big data, computational tools in general, as a step that is too big to take the first step. So like Liz said, my background is in neuroscience. So just to make sure we're on the same page and to introduce you to what I do, the goal of my lab is really to understand how microglia function during neurological diseases. So if you're not familiar, microglia are small cells that bind gelatinous material, literally taken from the origin, sort of the origins of the language. That's what it means. And recently, there's been an understanding that these are much more than just small cells that interact with things in the brain and support neurons. Although they do support neurons and they provide functions such as synaptic pruning. They're also responsible for sensing the central nervous system environment and responding to any damage, and they provide really the immune function, the primary immune function in the brain. And so you'll see them perform functions like phagocytosis, where they will engulf debris, they'll produce cytokines, and participate in inflammatory signaling. I bring that up because we'll talk about some data that we've worked with in microglial cells. But if the goal is to understand microglia, I also want to integrate these so-called big data strategies or bioinformatics strategies to supplement the traditional lab work. And I think that this is important because it can expand the reach of my lab and it can incorporate more students and give them a wider exposure to various ideas in the field and what I think the field will look like in their careers. So to do this, I want to think about first the NIBLSE Competency 1. And so the NIBLSE group came up with a set of competencies. And the first one is they said that students at the undergraduate level need to explain the role of computation and data mining to address both hypothesis-driven and hypothesis-generating questions in the life sciences. And with that, I think what we're doing is we're trying to address some misconceptions. And I want to talk about those misconceptions and how I've tried to address those misconceptions and how I've tried to get students to understand the importance of this competency and get them to competence in this. So the first one I want to talk about is that mindless data mining is a cheap trick, right? And I think we've probably seen papers before where they show a bunch of data and it says that things are different, but at the end it's hard to come up with a conclusion of what they actually mean by different and what the main takeaway is. And this leaves us exposed to this so-called type 3 error. I guess the lay term or the lay version of this is that there's a really high chance of a right answer to the wrong question. Statistically, I guess what that means is that you correctly reject the null hypothesis, but you do it for the wrong reason. Okay, maybe you have a random event that occurs to that leads to that conclusion. And so in bioinformatics, I think there's, at least at the undergraduate level, a high chance of this type of error because students want to start looking for differences. And they're focused more on, can I get a statistical difference, or can I get something that looks good or that is really interesting, and they don't necessarily think about the biology. And so what I've tried to do is try to really strongly integrate the biology of microglial cells or whatever model you're working with the computational or data tools that we have. The second thing that I emphasize is that the data that you get out is only as good as your experimental design, and that's only as good as your question. And that's true for any type of scientific data. But what I've seen, and I think is supported in the educational literature, is that students who interact with computational tools sometimes don't understand how to design an experiment well. And so the last one that follows from that is that curiosity and creativity are what's driving science. It's not the techniques, although the techniques are very important in that. At the end of the day, bioinformatics is a technique, just like any other technique that we have in our toolbox. And so how do we best apply that technique, and in what ways can we integrate that technique with what we already have in our toolbox? And this is a phrase that I've heard quite a lot when I talk about bioinformatic data. And so you'll get a heat map in a paper that looks something like this. And it can be a little overwhelming where things are changing. You have some treatment and a control group, and there's hundreds of proteins that might be changed. And I often hear this from my students where they'll say, that's really cool, but what does it mean? And so the question is, how do we make sense of all of these data? And what I've tried to do at the undergraduate level is to emphasize that these models or these type of data are a way for us to explore unexplored possibilities. So in his book, Rules of Contagion, Adam Kucharski to the fact that models allow the identification of unexplored possibilities that we may not yet have predicted, that may not be predictable by common sense. It's not a direct quote, but he talks about this idea that models and these large data sets allow us to identify what is possible. And so what I've tried to do is take this idea and integrate that into the undergraduate classroom, and specifically into lab courses. And so what we do is we emphasize making an observation. You gather data, you develop some predictive model, and then you propose maybe more experiments. And this is the process of science that we want to emphasize in all of our students, no matter whether it's bioinformatics or another field. What I think is important is that we emphasize where bioinformatics is useful. And specifically for the classes that I teach, I really emphasize this at developing a model. And if we can gather experiments, can we then use bioinformatics and computational tools to then propose other experiments? And I think that helps undergraduates decide where to put this. And so a lot of what I'll talk about is how do we integrate bioinformatic or big data ideas in with traditional wet lab experiments or wet lab exercises in the classroom. So the first way that I want to talk about this is what I do regularly in the classroom. And so I'll talk about the classroom first, and then I'll talk more about on the research side and how I teach my independent research students how to use this. So on the classroom side, I'll use tools like neuromorpho.org. And so this website and this database provides reconstructions of brain cells. It's a fantastic resource that allows researchers to database their reconstructions of neurons or microglial cells, astrocytes, any brain cell. And along with that, they have they have the original data files there. They also have data on the longest process length, the soma surface area, the cell volume, all of that ends up in this database as well. And it's freely available, and so you can download data from there. You also have published data where you have gene and protein data. And so you get some expression data, and what I do is I'll have my students pull this data out. So there's been a paper that's published, and they're required to make their data available. We can get that data. We may have to learn a little bit of computer code, and so we can use our other programs that other people in this seminar series have talked about to then process that data. Or sometimes you'll get an Excel sheet with fold changes or something like that. And there's a lot of emphasis on trying to understand and how do we deal with these types of data. For my students, what I do is we focus on getting enough so that we can work with it. And what I really want my students to do is integrate that with their student-generated data. So I want them to start generating data. Once they've generated that, we then compare that to data in Neuromorpho or to published RNA-seq data or proteomics data. So I'll provide one brief example of this from a project that a couple of my students did related to ethanol. So the students had a question about ethanol exposure, and they ended up measuring soma size. And so they were measuring the soma of the microglial cell. And this is a graph that they ended up making using data that they collected in the lab, as well as data that they took from neuromorpho.org. They ended up normalizing it to 1, and so that's why the soma size doesn't have a scale on there. But the power of this, I think, is that they were able to get access to this data. And specifically, this data was collected when we went online in the spring. And so the sample sizes were really, really low for our studies when we went online. And so they were able to go back to Neuromorpho and find studies that replicated or had a similar experimental design and integrate that with their data. And they could combine that to generate a graph. Okay, and so this is not a perfect graph, not a perfect design for this study, but the students were still able to engage with this and still learn that process of thinking through what is the experimental design, and then how do I take this together and analyze it. And so the gray and black are at 2 different time points. Along with this, they generated some data, and specifically it was from flow cytometry. And this is a graph of P2RY12 fluorescence in using flow cytometry. And they initially validated this using LPS stimulation. And this LPS is the immunogenic portion of E. coli. And it's often used with microglial cells to induce a pro-inflammatory state. And at this point, they were getting ready to then test and compare the expression of P2RY12 in LPS and in ethanol, like they had started to do with the soma size. And then we went online. And again, we went to the literature and we found that there were studies that had a very similar experimental design to what we wanted to do. And so what they could do is they could pull out RNA-seq data from various studies, and specifically we had 2 different studies that had a similar experimental design. So we pulled the data from that, and we were able to dig through and find proteins that we were interested in. And so this is not necessarily, I guess, the best use of bioinformatics, just scanning through and finding specific proteins, but it allowed students to get experience finding the data, processing it, and finding ways to then compare across studies and combine the data together. And so then out of this, they picked several different terms, or several different proteins, sorry, that were different. Okay, and they found differences. And then I had them go and do a GO-term analysis. So gene ontology is a database where we can define molecular functions and biological processes. And these terms are then encompassing many different proteins. So they put in their data into the gene ontology database, terms that they got out. And so then I said, okay, now what we can do is we need to then figure out what term do we want to follow up on. Okay, and so we take all of the data that we have, and then they decided to study microglial migration. And so when they looked at the microglial migration GO term, there were many different proteins in there, and a lot of them they hadn't yet studied, things like TREM2 and CSF1. Okay, and so I said we could look at other targets in this GO term to see what's happening with ethanol or LPS or whatever other stimulation we're looking at, or we could do a functional assay. And what they decided to do was a functional assay, and the data was called a scratch assay. It's a fairly simple assay. The idea is that you have cells growing in a dish, you take a pipette tip, you make a scratch down the middle of it, and you see how much of that scratch gets filled in over a certain period of time. And they found, and so they were able to calculate this migration index. They ended up not finding any differences, but I think, number one, that's okay in a sense, because it illustrates this process of science that sometimes you go through and you think you have the answer, and sometimes it's not there. And in some ways, I was, I guess, a little bit happy, because this provides a good opportunity for them to think about, is a scratch assay the only functional assay that we need to do to address microglial migration? And so by integrating these bioinformatic tools, what we were able to do is take data that they generated, compare it to the literature, combine that together, and then come up with a future direction for that study that they could then do another experiment and design another experiment for that. And this is how science is done. And so by integrating this, I was able to give them some exposure, and students start to feel like, oh, maybe this data isn't so daunting after all. So when we think about this idea of mindless data mining, and we want to keep away from just looking for whatever statistical differences are there, what I try to emphasize is, if we have some data, we can then take that and combine that with existing published data, whether that's morphological data, whether that is RNA-seq or proteomics. We can then integrate that with what we have and use that as a foundation for future studies. And I think this is especially important when we think about teaching students experimental design. And if we think about another couple of misconceptions that we want to address, is that we can't just randomly start looking for differences. We need to think about our experimental design and the question that we want to address. And we want to make sure that curiosity and creativity are driving our research, our experiments, and not just because we want to use a cool new technique. Okay, so the other thing that I want to talk about is our work with amyloid-beta and Alzheimer's disease. So just to make sure we're on the same page, I want to give you a little bit of background, and then we'll come back to some proteomics data that we have related to amyloid-beta. So in Alzheimer's disease, the amyloid precursor protein is cleaved. Normally, this is cleaved into fragments that are non-pathogenic, fragments like A-beta 40. In Alzheimer's disease, there is an accumulation of an aggregate-prone A-beta 42. And so this form builds up in the brain, and this builds up even before we start to see clinical symptoms and cognitive symptoms. So in the red, this is showing elevation of amyloid accumulation in the brain, and that often in Alzheimer's disease patients precedes neuronal injury and clinical symptoms. And so our question is, what impact does this have on microglial cells? And again, we're interested in how microglia are responding to neurological diseases. And so we're looking in the context of Alzheimer's disease, we're looking at the response to amyloid-beta 42. Now, we also know that in Alzheimer's disease, there's iron accumulation that is associated with microglial dysfunction. And so these are this is a published study that is showing ferritin accumulation in microglial cells. And what I want you to notice is that as we progress in Alzheimer's disease, we get ferritin accumulation, and we also get microglial dystrophy. So the microglia change morphology, and this morphology tells us something about their function. Okay. The big takeaway from this is that we see iron accumulation correlating with microglial dysfunction and amyloid pathology in Alzheimer's disease. And so what we tried to do is we tried to come up with a model to study chronic iron accumulation in microglial cells and whether or not that impacts the response to amyloid beta. So if we have amyloid beta that is released into the extracellular space as a result of Alzheimer's disease, that will then be sensed by microglia. This is part of their job, is to sense and respond to changes in the environment. And our question was really, in this cell, in the microglial cells, impact the response to this amyloid accumulation. So the design for our experiment was that we added iron to our microglial cultures, and for these experiments, we used an immortalized mouse microglial cell line. We added it to our cultures for 48 hours. We then stimulated these cells with amyloid beta. And our question is, is iron altering this response? And so this is a 2x2 factorial design where we have a control group, iron stimulated cells, and then we have both treatments together. One of the first things that we found was that iron actually altered the ability of, or altered the phagocytic capability of these cells. And so what we did is we can put fluorescent beads onto the microglial cells, and what we found is that both iron and amyloid beta stimulation increased microglial phagocytosis. So the big thing that we took away from this is there's something going on, and we thought this is great. You know, I thought this is good data, and we have a mechanism. We can start working with this and picking apart the mechanism. And one of the first experiments that we did to address what I thought was the mechanism showed no difference. So we did another experiment to test it in a different way. And we started looking at it and figured out, okay, that's not the mechanism. And so we did a proteomic study where we wanted to then find out what differences exist. And so what I encourage my students to do is think about, we can use proteomics to open the doors to think about alternative directions. And so rather than kind of blindly looking around, or maybe even in a somewhat targeted fashion, looking at different functions, let's use these big data tools to then try to come up with a hypothesis of where to go next. So what we did is we did this proteomics study with the help of the UAMS Proteomics Core, and what we found was that we had differences in both iron, amyloid beta, and amyloid beta plus iron. Okay, and I'll talk about more the results of that in just a minute. But for my undergraduate research students, what I have done with this bioinformatic data is I've first asked them to evaluate the model. Okay, and so we have this model where we say we think we're modeling what's going on in the brain, where we have iron accumulation and amyloid beta. And so I asked them to answer questions like, does the proteomic analysis that we have, the data that we have now, support our model of iron dysregulation in microglial cells? Is there agreement with published studies? We're using an immortalized cell line. That's not a brain cell in a human Alzheimer's disease patient. We're miles away from that. But is there agreement between what we found and what our model is and what is published in the literature? And so the first thing I asked them to do is to evaluate the model. And so we got a bunch of Go terms out of this, and we started comparing amyloid beta stimulated cells to iron and amyloid beta stimulated cells. So the thing that's changing here is iron, and we started looking at these Go terms to see, are we actually getting iron dysregulation or metal dysregulation in these cells? So we found Go terms like metal ion binding was changed and sequestration of ions. And as students start to evaluate these Go terms and think about what does this mean in the context of evaluating our model, they then get exposure to these ancestry trees in Go term analysis. And so we start to then push them to get familiar with the type of data that they can get out from things like Go term analysis. And I really push them and encourage them to think about these things, because as they go forward in their careers, this is a very useful tool. And so in this context, it's a way for them to think about it in somewhat of a safe space where we can evaluate the model, and whatever they find, we can move forward with. But I want them to be familiar with these tools at some level before they move on out of my lab. So the first thing that I have them do is evaluate the model, use this proteomic data, and think about, is there a way that we can validate our findings? Okay, we have experimental data, and how do we then incorporate bioinformatics, or in this case proteomics, into our studies? The second thing that I then want them to do is to select a target, and this is where I then use a lot of bioinformatic data in larger class settings. And I'll ask them, if we have this big data set, how do we go about picking a target? And once we pick a target, what do we know about that gene or that protein? And then is there a way that we can use that as a rationale for future studies? And so early in the semester, I have them pick this protein or gene, and then what we do is think about, how do we then design an experiment to test the role of that gene or protein? And there's a couple of different ways that I approach that. The first thing that I emphasize is that we need to lean on our experimental design. Okay, we had an experimental design where we had 4 groups, where we had control, iron, amyloid beta, and amyloid beta plus iron. And in our Go term analysis, we started looking through some of the Go terms that were commonly changed across all groups, and we started to find some proteins like arginase and ferritin that were changed in all of these Go terms, or in many of these Go terms. And so I encourage them to think about, we've got an experimental design that allows us to identify proteins that are potentially important in many different pathways. So maybe that could be a target. This Venn diagram was made by Yasir in the bioinformatics core, and when he first sent me this Venn diagram, I got really excited. And so what I want you to notice is we have all of the differentially expressed proteins in the iron, all the differentially expressed proteins in A-beta samples, A-beta stimulated samples, and then all differentially expressed proteins in iron plus A-beta. The thing to notice is in all of these groups, there was only one protein that was different. And so this makes a pretty good rationale for picking it as a target. And so we looked at, what is this protein, and how do we then start to interrogate this? Okay, and there's a couple of different ways that I encourage my students to think about once we've picked a target, what do we want to do with that? The first approach is that you might have several student groups, and I might have 2 or 3 different groups in any given semester, depending on the size of the lab. We can then, as a class, decide, do we want to select and optimize a specific assay? And then do we want to pick all different genes to modulate? Okay, for instance, if we pick for microglial cells, one of the most common assays that we do, one that I talked about earlier was the phagocytosis assay. So do I want to, or as a class, do we want to pick this assay? Do we want to really optimize it so that we all are really good at this technique? And then group 1 will pick one protein, group 2 will pick a different one, group 3 will pick a different one, and then we try modulating this protein to see which of these impacts phagocytosis. One of the big benefits is this allows for a lot of choices for students. They get to pick the assay as a class that they do. They have a choice of optimizing their experiments, and then they get a choice in the gene that they want. Okay, and they can form groups based on what gene or protein they're interested in. A big drawback to this is if you've done any work with genes or proteins, modulating these things can be tricky. And so transfection efficiency or selection of antibodies can be difficult. And so some groups get stuck at simply knocking down their protein or transfecting in and trying to overexpress a protein. And so I've generally tried to stay away from this, especially for groups, unless I have some students who really, really want to do this, which I haven't so far. But I generally try to steer them away from this approach simply because I'm one person and the manpower for this is difficult. The approach that I've used more often is I might have several groups, and together we select a protein or a gene to modify and modulate. Okay, and then we pick our favorite way to modulate that target. We use siRNA quite a bit, but you could use blocking antibodies, CRISPR, any of these strategies. So once we select that, we then, as a group, are all working on one protein or one gene, and then we perform different assays. So maybe group 1 does a phagocytosis assay, group 2 measures the production of reactive oxygen species, group 3 measures activation markers in these cells. And so this is the approach that we've used most often, and I'll share with you what this looks like for a specific protein, for the one protein that we identified in our screen. And a big benefit of this is it allows a lot of prep because I know what siRNA I need to get and what materials I'm going to need at the beginning of the semester. A big drawback or a potential drawback of this is that this can be a lot of work for the instructor and for the students depending on the assay requirements, because they're going to have to optimize this and sometimes depending on how long it takes you to pick the target and optimize and teach them the transfection or the antibody treatments, that can push you further along in the semester. But either way, at this point, once we've picked a target, they've really been exposed to a lot of these tools, things like GO-term analysis, things like differential gene expression, and some of the statistics that go along with that they haven't really, at least for my students, they haven't covered in their biostatistics course. So we selected a target, and we selected this one protein that was differentially expressed in all 3 of our groups. And that protein was NIBLSE C1, or normal mucosa of esophageal-specific gene product 1. Okay. And so once we picked this, the students then had to go figure out, what do we actually know about this? Do we know anything about this? Before we start trying to just do things in the lab, we want to figure out what things we already know about this protein. They figured out that it's associated with the GO terms for the electron transport chain, proton transmembrane transport, and response to bacterium. And so at this point, they're looking at me going, okay, now what? It's like, keep going. What else do we know about this? So as a background, this protein was identified in 2000, or first reported in the literature in 2002 in an esophageal squamous cell carcinoma patient. Since then, it has actually been identified in gene microarrays in a patient, in an Alzheimer's disease patient, where they stimulated the cells with amyloid-beta, and it was up 16-fold in that paper. And when my students came across this, they were like, why has nobody looked at it? It's like, this is a lot of data. And so digging through a microarray from 2005, we actually found that this protein had been identified before. And so we kept looking of what else do we know about it. We found that upregulation may be protective in certain inflammatory diseases, peripheral inflammation, so not in the brain. And we found that the parent gene of this protein will generate several microRNAs that are linked to inflammatory conditions. And so we then asked the question, in our microglial cells, what effect does this protein have? And again, all of this up until this point has been outside of the wet lab. And so these students have had to think about, how do we select a target? And in this case, it wasn't real hard because we had one protein, but how do we select a target? How do we dig through all of this data where we have several thousand proteins that are different? And what we found is that NMES1, when we block it, we actually get an increase in activation of these cells. And so I'll show you this one piece of data that the students generated, where when we use siRNA to knock down expression of NMES1, what we found is that we get an increase in microglial markers of activation. And this is specifically CD68. And we found that amyloid-beta, as expected, increases CD68 expression. And so that's indicative of the pro-inflammatory nature of amyloid-beta, which has been well documented. What we thought was particularly interesting is when we transfected with our siRNA for NMES1, we got a further increase, which would suggest that NMES1 is somehow providing a brake onto the inflammation for these microglial cells. And so by doing one experiment, students have now had to work through all of this bioinformatic and proteomic data to get to a point where we do an experiment and we can generate some data. And this is not a trivial task for some of these students. So I'll show you all of the data that we have related to this, but just to illustrate that this approach has allowed students to generate their data and gotten them interested so that they find it worth following up on. Okay, and so here's our model that we're building. So if we come back to this idea of addressing this core competency, if we want students to explain the role of computation in data mining, and both to address this hypothesis-driven approach as well as a hypothesis-generating approach, we want to address misconceptions. And what I try to do is make sure that they understand that bioinformatics is a tool. It has a place in the research lab. And particularly, I do that by trying to really strongly integrate it with the wet lab experience. And so hopefully going forward, they then have a foundation to start thinking about, if I get a large data set of RNA-seq data or proteomics data, I'm more comfortable learning the computation tools because I now understand the power of that. And that's been the goal so far for integrating these bioinformatic tools into the classroom and for my independent research students. So before I wrap up, I want to think very briefly about barriers to entry, because for undergraduate students, there are several barriers to entry that I think drive some of these misconceptions. Number one is faculty competence, and that's probably a whole seminar series on itself on giving the faculty tools to do this. But I have to know that I have limitations as far as bioinformatic tools, and as new things come out, I need to keep up with that, but I also want to design ways where students can use these bioinformatic tools where they won't be hampered by my ability to keep up with the literature, things like that, if I fall behind or something like that. And so another barrier that often happens for undergraduate students is they look at it and they say, I can't code. And so it's a struggle to overcome this barrier, because at some level you need this to work with proteomic data or gene expression data, things like RNA-seq. You need to be able to run a code in R or use some basic command line tools. And so to address this barrier to entry, what I really do is try to teach the rationale for using these tools. And so if we can develop a scientific rationale using bioinformatics or computational tools, that's really powerful. But we don't necessarily, for that, need to know how to code perfectly, okay? And then I encourage them to learn while they're learning these wet lab techniques. And sometimes the coding part of it can be very discouraging. If you've got a period on line 100 and whatever of your code, it can be frustrating when you're trying to troubleshoot code, especially if you're using somebody else's tools. And so I encourage them, let's learn some of these techniques so that we can kind of build some momentum as your tougher tools, or what in their mind are tougher tools in computer coding and things like that. And then I also encourage them, is there a way that we can address novel questions using existing data? There's loads of data out there that are published from RNA-seq data and proteomics data. Can we then dig through that data and find something new? And I remember sitting in a meeting, I think when I was in graduate school, where a researcher got up and said, we have some really cool data. He walked through a lot of their experiments and so the RNA-seq data, and he acknowledged, he said, we have an incredible amount of data that we have not yet completed all of the analysis. And he made that publicly available on his website and encouraged everybody at the meeting to go look for interesting things that they find in that data. And I think the same is true for particularly for undergraduate students, that it doesn't necessarily have to be novel to the scientific community. It can just be novel to them. And that can be a powerful teaching tool. I think another big barrier to entry is that bioinformatics seems incredibly overwhelming. When you get 15,000 proteins in a proteomic data set, you can think, how do I even begin to analyze that data? And so what I really try to do is I try to start small and focus on learning. And it's even things like matching genes to proteins. So if we know that we're measuring a protein in a cell, if we want to find RNA-seq data related to that, the gene name may not be exactly the same as the protein name. And sometimes there are many different protein names, sometimes there are many different gene names. And so starting small and just thinking about, what are all the possible options for protein and gene names, and can I go find different studies that address a similar question? And a lot of times we'll start with preprocessed data. We'll pull an Excel sheet, and the overwhelming part is that there's 10,000 lines on your Excel sheet, and I try to get them to understand that you can deal with that data. And then once you're comfortable with that, we'll take another step back and think about, then how do we generate that Excel sheet? How do we generate a differential expression or a log-2-fold change or something like that? Okay, but the overwhelming aspect of bioinformatics, I think, can be addressed by starting small and letting students focus on just a little piece of this at a time, and then building slowly over time. Although in 4 years, it's hard to go too slow. And then another big misconception that I've faced and that provides a barrier to entry is that a lot of students don't necessarily view big data or computational tools as, quote, real science. And this can provide a barrier because they want to do real science. They want to get their hands on it, and they want to be in the lab, and they want to look like a scientist and feel like a scientist, and they want to work in the hood and make a mess and wear a lab coat and all of this. And so what I try to do is build toward competence in understanding the role of hypothesis generating data and hypothesis addressing their hypothesis using these computational tools. And I think there's been a temptation for me to use data that I'm sure is going to give them interesting results so that when we're working with this, they can learn the tool and all of that. But I think sometimes that has driven this misconception that it's not real science because it's too perfect, it's too easy. And so if we can move away from really nice, clean data into, quote, real world experiences and real world data, where sometimes it's messy, sometimes people's code doesn't have comments in it, sometimes people's RNA-seq data has missing values and things like that, then we can start to work through the process of science where they have to troubleshoot and where they have to think about how do I deal with some of these challenges that I will face, whether this is a computational or a bioinformatic tool, or whether this is something in the lab. The process, ideally, is the same. And so this is how I've fought these barriers, or tried to overcome these barriers to entry and drop those so that students understand the value of computation and the value of bioinformatics. And once they understand that, hopefully they're interested enough to then go forward and pursue this in their future careers, or at a minimum, they're comfortable that if they're asked to do it, or if they see the need to do it, they understand that it is accessible to them, that the tools are out there, that they can learn it. So I hope that stimulates some ideas and discussion, and I'm happy to stick around and answer questions or have a conversation about any of this, but I'll wrap up here. So thank you for your attention, and thanks again to Liz for putting this together.
David, thank you very much. I invite people to ask Dr. Donley questions. We actually have a question. One of the audience members would like to know, were the students' work explained in this talk integrated into a single course for one semester, or were they spread over multiple courses for multiple semesters, or evolved into summer research? What was the time span?
So for the class part of it, so the ethanol data that I talked about at the end where they're measuring CD68, that is in a single semester. I have students in the summer and throughout the year who will work with me as independent research students. So that occurs over a longer time frame. And so some students, when they come in, I work with them over multiple semesters. But generally, I think everything I talked about in here was over a single semester. And so trying to pack all that in is tough sometimes. But as a background, the course that I do this in, it's called Neuroscience Research. The entire goal of this is to give them at least a semester of research experience before they leave. And so when they come in on day one, we go over the syllabus, takes me 20 minutes or so, and then we start learning to culture cells. And then, the next week, I expect them to be fairly competent at culturing cells. They start designing experiments and reading the literature. And generally, by about midterms, they're generating their own data. At least that's been the goal most semesters. But yeah, so it's over one semester.
Kim had another question. She was wondering if you had any particular prerequisites for the research projects and classes.
So let's see so the neuroscience research class that I talked about, the prerequisites, they have to go through genetics. So they have to be at least a junior, I guess, in our curriculum to get there. I've done some of this at a little lower level with as low as freshmen. And then in we did a little bit of it in a neurobiology class, and the prerequisite for that was cell biology. So again, they were mostly juniors. So most of this has been done with upper level students, just because trying to teach a freshman all the genetics that they need to understand that and not just view this as magic is tough. So if you have ideas on how to bring that to a different level, I would love to hear it. But yeah, for what I talked about, all of these students are juniors to get through prerequisites like cell biology and genetics.
I wanted to ask, sort of in a perfect world, are there some additional things that you'd like to be able to do with your students that haven't yet been able to either work into your curriculum, or there have been other barriers that prevent you from some of the other things you might have on your wish list?
Funding. I think that's always a big issue is, we're working primarily with student lab fees, and it's hard to balance research funding with class fees and things like that, because I want to give the most number of students their opportunities to engage with this. And that's hard because you can do things in a shorter amount of time if you have this crazy large budget, but that's not real life for most schools in the United States. And so I think getting students to understand that is also something that's really difficult, but also very useful, because they have to understand that we can't just go out and buy a bunch of antibodies, unless we have the funding to do it. And so I think that's been an issue that is not easily solved, with a seminar or something like that. I think another big barrier is once students understand the importance of this, passing them off to someone else to teach them the computation or something is difficult, because it requires that your curriculum is really well streamlined. And in the higher education setting, that's very difficult to do, is to make sure that we allow the most number of students to get access to these classes and to these experiences, but also require enough prerequisites that we know what they're coming in with, so that we can kind of hit the ground running on day one. And that's been a big challenge from the faculty side, and a barrier to some of this is when we want to teach something, we also need to make sure that our class is all the way on board first, and we don't leave behind students who especially are at risk when they come into our classroom. And so that's been a challenge, because this is one semester. Ideally, this is a 3-semester sequence where they begin learning these things or something like that. So I don't know if that totally answers your question, but those are the two that come to mind immediately.